Hello, this is Mrs. Rowe, and welcome to our Learning Biology from Home series. And today we are going to wrap up our cell transport and how cells are able to move materials that they need in and out of the cell, whether it's materials that they need to take in or materials that they need to get rid of, waste products, because waste products can also be harmful to the cell. So, how do they get these things across these bilipid membranes? That's what we want to be looking at today. So, we're going to start by talking about active transport, which is where we left off the last time we spoke. We talked last time about diffusion mountains, so hopefully you have your notebooks ready. You hopefully paused the video and drew your mountain. This is a great way to remember why the molecules move the way they do. So passive transport, just a quick review, you're going from a high concentration to a low concentration. It is driven by this difference in concentration across two spaces, usually across your membrane. If there's a change in concentration, simply the motion of the molecules, that's what powers it. Molecules are in constant motion, and this constant motion, as they're bouncing off of each other, they're going to just continue to spread out within the space that they're in. So if they're going to just, that's what's going to drive them, and it's kind of like dropping a ball off the top of a mountain, and it's just going to go on its own without any energy on your part once it gets started. The bigger the concentration gradient, the faster it's going to move. And we say it is going down the gradient or with the gradient when things are moving passively. So those are some words to watch for. Temperature, obviously, since it's driven by the natural motion of molecules that they all have, that kinetic energy that they have. So temperature increases kinetic energy. Temperature will increase the rate of your diffusion. Charge has to do with polarity, because as you've seen yesterday, polarity is going to influence what can and can't cross that nonpolar lipid barrier. And then also, obviously, diameter. Larger molecules are going to get slowed down and have a tougher time getting across. And that's where sometimes we get into helper proteins, facilitated diffusion. So diffusion is simple motion across the membrane, such as CO2 and oxygen. Osmosis is specifically water diffusion, and facilitated diffusion are those things that have a charge differential, or are too big or need some help to get across. Well, today we're going to start talking about active transport. Oh, and let me finish that our gas exchange, CO2 and oxygen, and obviously movement of water, those are all key important things. Even our capillaries, when they are delivering sugar, we've had our meal and sugar is in our bloodstream. That sugar is then going to naturally diffuse into our cells where there's a lower concentration of sugar. So many, many important things are going to go both in and out of the cell through passive transport. But today we want to start moving over onto active transport. And today we are going to be going up the mountain. So if you want to pause and get ready, this time we're going to have our little person. I found some little clip art to put for our little people. And you're at the bottom. You're at a low concentration, but you need to stockpile. You need to go up the mountain. And as you can see, that's going to take some energy. That's going to take work. If you need to move this molecule to a higher concentration, you're going against the flow. I think of the flow like a river. If I'm sitting in a river in a canoe and I'm going with the gradient, with the flow, I don't have to paddle. It's just going to carry me downstream. But if I got to paddle upstream against the current, by diffusion, everything's moving in this direction. I want to paddle against the current. I got to work harder. Energy is required for active transport. We're going against the gradient. And the purpose of this is simply to stockpile. So active transport is about stockpiling materials, where you want an increased concentration. And there's multiple reasons why you want to do this. This is how our mitochondria work to recharge ATP. We're going to create concentration gradients, and then when we release that stockpile, it's going to flow down all at once, and that's how some of our pumps work. We're not going to get into the details of that. So that's how we maintain water balance with pumping our sodium potassium pumps. Notice what active transport, those proteins that are involved are called pumps. So we have our active transport. We have our hydrogen ion pumps for ATP. We have our sodium potassium pumps for maintaining homeostasis in our cells. So even though we're stockpiling, it's still sometimes about maintaining homeostasis. Active transport is how plants are able to pull minerals in that they need from the soil because these minerals are hard to cross the membrane. So this is how they can get them across. It's how they can get transfer the sugar they're making into photosynthesis into the phloem or the tubes that are going to deliver and transport that sugar through the plant because you're putting it into a higher concentration. So to get it into there, you've got to stockpile it. You're going to need active transport. So active transport, we use it for a muscle contraction, active transport with ions, nerve impulses. Many of the things that we use and require active transport. It is just as important for cell homeostasis and survival as passive. So they're both equally important. So you're going against the gradient, you're going from a low to a high concentration, and they often involve a protein to carry them across. So we're actually going to, there's a couple of different kinds of active transport. So let's talk about those briefly as we are talking about active transport. We have our pumps, such as the sodium potassium pump. We have things such as um, exocytosis and endocytosis. And those are a little bit different in that you are not, I think I spelled that wrong. Go back and fix that. But those are a little bit different in that you're creating membrane to create these little vesicles to move large quantities. Again, you're stockpiling. You're taking a whole bunch of insulin. You're taking a whole bunch of whatever the molecule is that the cell's producing, and you're putting it into a vesicle so that you can move it around the cell or move it out of the cell. So let's just fix our spelling here. Computer's a little laggy. Okay, so those are some examples of active transport processes, whether using a sodium potassium pump, hydrogen ion pump, or an ATP pump, however you want to refer to that, exocytosis, endocytosis. So let's take a quick look here at our membrane and let's talk about how this is all happening. So don't forget to pause if you need to finish your diagram over there. So here's our bilipid membrane with our phosphate heads that are polar and our non-polar tails in the middle. Well, simple diffusion. This over here might be oxygen or CO2, and I'm not going to worry about that is going across because these are small molecules and they are non-polar. So to be able to get across over here, they need to be small and non-polar. I know I got another spelling error. I'm just going to leave it. So small and non-polar will be able to diffuse directly across. What if you're large? What if you are polar? So things that are polar, so larger, not huge, because huge, when they're really large, that's when they're larger than the protein itself, then we're going to need something like exocytosis. So larger or polar or ions. So things with a charge. How do we get those across? That's where we need a helper protein. And that's where the transport protein. So these have multiple names. So this could be called a channel protein. It's just an open channel. This might be one way that water comes across through something like osmosis. Osmosis could do this to get water across over here. Often this one looks more like an ion channel, so maybe you have some ions that are being brought across passively from high to low concentration. All of these go from high concentration to low concentration. Notice these channels can be gated, that it can close, it can control how many are coming across. So you can have gated channels that can control it, you can have open channels, but they're all basically called, there is slight differences, but they can be called carrier proteins, transport proteins, channel proteins. Um, So you can see we've got lots of names, but basically a protein that helps them get across. On the flip side, active transport, we're going from a low to a high. Notice there's only two triangles here and there's several here, but we want to keep stockpiling. We want to keep bringing them over. Um, what you want to do then is you're going to use an active transport protein where they're going to load into the protein, and then you need some energy. I think of this like the doors at the grocery store, those automatic doors where you open up and you kind of wait and then it detects you, and then some electricity will open the door and allow you in. But if there was no electricity, that door would stay shut if the power were out at the store. And it's kind of the same way. Think of it as an automatic door. You're not getting across unless we have power to allow you through, and it's going to keep using it. It's that kind of like a pump. I also think of it as like a sump pump in your basement. If you have water in your basement, you turn on the sump pump. That takes energy and it's going to pump it to the out of your basement. If the pump shuts off for whatever reason, you lose power, that water through diffusion is going to come back again. It's going to go back from high to low. So you got to keep pumping it to keep it out. Um, and then we have the other two processes. What do you do if you need bulk transport, like a whole lot of insulin protein molecules? They're not going to fit through these little molecules by themselves. What if you're like an amoeba and I'm consuming an organism? How do I do bulk transport of things? A starch molecule, those are really big. How do I get things across membranes when they're too big to use one of these processes? So that's where we get into the endo and exocytosis, which are also considered active transport. You're producing membrane in the process, so any kind of manufacturing requires energy. And so I have a quick little video clip for you, and then that will be it for today. So this is an older video, but in fact, it's actually one of the best animations I have found. I have not found anything yet that I like better. Um, so we're just going to watch this. So I've skipped ahead in our video. This is the endoplasmic reticulum here. This is a mitochondria. We're inside the cell, and this yellow stuff on the right is the Golgi apparatus. And so think of this like a pancreas cell producing insulin. So it's rough ER, and those ribosomes are making some insulin, and then they're going to stockpile them all together into one vesicle. And you're going to see how it then moves through the Golgi to be packaged, and then through exocytosis is going to be released from the cell. Here we go.
After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane.
Now this
As
is the
membranes
part.
merge again, the contents leave the cell without actually crossing the plasma membrane.
See your bilayer? Isn't that cool? And it can go in the other direction if you want to take things
This
in
process
bulk.
can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
What did you think? Endo and exocytosis, and seeing how those vesicles form from that membrane as they pinch off. I think that's really cool. And that's actually how things move from organelle to organelle also. So that, in a nutshell, is active transport. We are not going to get into how the sodium potassium pumps work. We're not going to get into details. Just be aware that active transport is for stockpiling. It's still important. And one last thing to pay attention to, if you look at this, passive on the left, active on the right, obviously passive doesn't always need a protein. It can be simple diffusion. But what do they both have in common besides the fact that they both help the cell maintain homeostasis? They both can use carrier proteins. One uses proteins without energy to let it go with the gradient. One is going against the gradient, which needs energy, but both process can use those carrier proteins or transport proteins. Okay, well, I hope that was helpful for you. If you have any questions, please let me know. Until next time, have a great day.